Welcome back. Give yourself a few seconds, mentally kind of arrive. Welcome back. Glad you're back. We're going to continue, maybe. There we go. Sorry, I thought it crashed. My crash screen came up. We're going to continue talking about phylogenies and the phylogenetic theme. It's kind of everything that we've been talking about so far. But you just watched a video on the concept of horizontal gene transfer. That difference from the vertical transmission of genes, where genes can get transferred between phylogenies, not from parent to child. And this is particularly interesting and relevant when we talk about bacteria and when we talk about antibiotic resistance, since bacteria are able to transmit those genes horizontally. Couple of other, so there's that link. The top one links directly to the organization that produced that video, and the bottom one is the YouTube link, so you can go to either one if you would like to watch that video again. Horizontal gene transfer, that's pretty crazy. That's definitely a variation on the phylogenetic theme. Another variation on the phylogenetic theme, if we look at sunflowers, we can build a tree that's pretty similar to what we're used to. We have this nice simple tree with these 5 sunflowers. That's cool. But wait, there's more, right? If we add in these specific 3 additional species of sunflower, the picture gets a lot messier, because what we find out is that each of these different species, each of these 3 are different species, originate from hybridizations of the same 2 species of sunflower. This is another case where we can get a phylogenetic tree that looks more like a net than it does a tree, is with hybrid speciation. So the hybridization of these 2 sunflower species has produced 3 separate species of sunflowers. This is not at all uncommon in plants to have this kind of hybrid speciation. It's less common in animals and particularly in vertebrates, but in plants this happens a lot. And so you end up with a tree that's not nearly as simple as sort of our basic foundational understanding of a phylogenetic tree. Other weirdness that can happen is if you're looking at specific genes. Oh, that's not what I meant to do. If you're looking at specific genes, in this case, this super awesome drawing that I did up here in the corner, those are aphids. And if you look only at the gene that encodes something called, oh, I don't have an orange, encodes something called the carotenoids. The ability to produce carotenoids, and carotenoids are like orange-red pigment. Most animals can't produce their own carotenoids. They have to eat stuff. They eat stuff that has carotenoids, and then they incorporate that color into your body. At the beginning of the semester, I showed you some of the lizards that I study, and they have those orange stripes. So the birds that you see my lizards are a bad example because it's not carotenoids, but the birds that you see, almost any bird that you see that's red or orange, they have to eat other things and bring those carotenoids in through their diet. They can't synthesize them themselves. Aphids are kind of cool and different. If we look at the tree, right here, this group, these are aphids. Okay, that's cool. You probably don't recognize the other scientific names. I didn't. But all these others, these are fungi. All of these species are species of fungus. These are all species of fungus. Oh, that's weird. So we have aphids, an animal. Their clade is nesting in the middle of a clade of fungus. What? Well, the reason that that's happening is because we're just looking at the gene that gives aphids the ability to synthesize carotenoids. We know aphids aren't fungus. What happened was somewhere along in evolutionary time, horizontal gene transfer conferred the ability for aphids to produce carotenoids, and it came from fungus. That gene came from fungus. So this is horizontal gene transfer from fungus to aphids. That's wild. That's so cool and crazy. So aphids got their ability to synthesize carotenoids from fungus. That's the only way to explain how this gene from aphids is nested, we call it, within a fungus clade. Crazy. Next, we have this warbler, this bird here. This is a warbler. And a gene that's, oh, did it go away? A gene that is really, really commonly of interest is a gene called the cytochrome B gene, and it's in the mitochondria. It's a section of the mitochondrial genome. If we look at the cytochrome B gene, this is the same species. What we see is that birds that live in Mexico, for this specific gene, remember this is all the same species, birds that live in Mexico, their cytochrome B gene nests together, whereas birds that live in the United States, their cytochrome B gene nests together. So the ancestral history of this cytochrome B gene, we have the origin back here, right? We have a Mexico clade and we have a USA clade. And what this is emphasizing is that, like we saw with the squirrels and like we saw with the aphids, that we can create a tree, a phylogenetic tree, from things that aren't organisms. We can create a phylogenetic tree from genes, and that gives us the history of that specific gene. I don't know why. Oh, I remember why. I did this. And a really important thing to understand is that organisms, one of the major ways organisms evolve is through the duplication of genes. So different organisms have different genome sizes. One of the major ways genomes grow is through duplication. So if we have, each of these colors is just representing an imaginary gene. 1, 2, 3, 4. We have 5 genes in our imaginary sequence here. And remember, each gene is made up of a lot of bases. Over the course of time, let's say that pink gene gets duplicated. Now we have 2 copies of pink gene in our genome. Pink gene makes pink protein. But now we have 2 copies of pink gene. So the pressure on the second pink gene to consistently make pink protein exactly correctly is released. Because the gene 1 is making pink protein. So it's making what the body needs to survive. What can happen is that now this gene that's been released from that pressure will mutate. It'll accumulate changes in its genetic sequence. So maybe it doesn't make pink protein anymore. Maybe it makes pink 0.0 protein. It's a very similar protein, but not exactly the same. This is an important concept in evolution, is that very frequent gene duplication, and that duplicated gene is released from the pressure to produce the exact same protein. So we can have changes in that genetic sequence, and those changes can produce slightly different proteins. Oh. How do I get rid of? Oh, okay. Wow. I was just talking to the group that I met with earlier today and saying, I still have not figured out how to work Keynote. What? Oh my gosh, guys. Alright, let's just go here. Ah. How do I erase that? Okay, hang on. I'm going to figure out how to get rid of that. I don't know why it's still doing that. Okay, hi, I'm back. Sorry about that. This gives rise to different types of descendant genes, I guess is kind of how you can think of this. Orthologs are genes that are inherited in the way that we're used to thinking about the inheritance of genes. Gene A is present in the common ancestor, and when those species speciate, gene A is present in each of those species. So it's present in species 1 and is present in species 2. Those genes are related to each other by common ancestry. So orthologs or orthologous genes are like what we learned about with homologous structures. They're related to each other by common ancestry. The other thing that can happen is if we look at this image and we imagine that at the pink point we had a gene duplication. That means that these 2 genes are experiencing different evolutionary pressures. So the red gene come along and it's under pressure. If it's the original, it's under pressure. Let's see, what do we want to call this? Let's say we'll stick with gene A. Gene A is under pressure to continue to produce protein A. So it's going through its evolutionary history, and it's still got to produce protein A. But we've duplicated that gene, and now maybe we'll call this gene A.1. It's going to start out identical to gene A, but over time it's going to experience different evolutionary pressures. So over time, what might happen is it might start producing a different protein if it experiences changes in its genetic sequence. So gene A and gene A.1 are related in a different way. These are paralogs. These genes are paralogs. And the way that I remember this is there's a pair of genes. Paralogs start with a duplication event. There's a pair of them. And because we go to gene A and gene A.1, then they can diverge. They can become different. They can possibly produce different proteins. Likely very similar proteins, but over time, they can produce different proteins. So what can happen is that the way that these paralogs evolve and change can be different for different species. So if this is species 1 and this is species 2, those species are going to experience different evolutionary pressures. So those two genes that exist because of a duplication event, they're similar because of a duplication event, not because they come from a common ancestor. They're similar because of a duplication event. They can experience different evolutionary pressures, and those different evolutionary pressures can cause them to evolve in different ways in different species. An example of duplication leading to, this should extend all the way to the top. An example of duplication leading to different genes and contributing to evolutionary change is hemoglobin. The hemoglobin molecule that we talk about the most in humans is 4 different polypeptide chains. 4 different polypeptide chains, that's 4 different genes. The actual hemoglobin molecule is the product of multiple hemoglobin genes. How did we get those multiple hemoglobin genes? We got them through duplication events. In this tree, each of the branches, is a duplication event. A duplication of the original globin gene. So this is a tree of the globin gene family. Here, this branch is myoglobin. Myoglobin is active, you may not remember, in your muscles. And it is a single polypeptide chain. Each of these branches is a gene duplication that led to new globin genes. This part is the alpha globin family, and this is the beta globin family. And what I'm going to do with this tree is I'm going to step you through how this tree, which I personally think is confusing and hard to read, understanding what's going on with this tree, research backs me up on that. How does this tree correspond to a rectangular tree? And I color coded this all for myself, so hopefully this will work. Okay, so starting here, here is our first branching. What that would look like in a rectangular tree is we have the root here, and we branch here. So my pink dot is going to go right there. We've branched. So that looks different than the sort of V tree. One of these branches goes all the way to present without anything branching off of it. So that would be that. That's myoglobin. I'm just going to put myo. This other branch here comes here and branches again. I'm going to use a blue dot for that. So we know that we come out here, we got to come up and we're going to branch again. Blue dot is at that branching site right there. Okay? We can follow one of those branches from blue up here and see we have another branch point. I'm going to do an open pink circle right here. That's another branching point. So we follow this up and we're going to branch again. Here's my open pink circle. Then we're going to follow, okay, one of these branches goes all the way to the top of the tree and one of them comes out and branches again. So one of those is going to go all the way up to the top of the tree. One of them is going to come up here and branch again. I'm going to use a blue triangle to represent that. Here's our blue triangle where we're branching again. Boop. Cool. You'll notice that one of those branches goes all the way to the top of the tree. So once again, all the way to the top of the tree. And then we come out here to another branch point. Okay, up. We have to branch again. Gonna use, what did I use for that? Orange. Gonna use a star like that to represent that branch point. Okay, one of those comes up all the way to the top of the tree. That would be this one right here. One of them comes up here and branches again. Okay, so now we've gone from blue dot all the way up to black dot. Remember that there's another blue branch here, and I've made my tree too big. There's another blue branch that comes up from blue. So that's, we go whoop, up. Okay, and we go up to another branch point. I used yellow dot here. We're going to branch. Whoop, branch. Okay, branching. Now we're going to follow one of those branches up right here. And it comes to another branch point. I'm limited in my colors here. We're going to represent that with a pink square. I apologize, I'm squishing the tree. Oh, we're going to branch again. Here's the pink square. And then we can follow, we have one branch that goes all the way to the top and another that comes to another branch point. So trees don't typically wiggle like that, I apologize. This is the one that goes all the way to the top. Then this one comes to another branch point. We branch again. I did a double circle here. Circle with a dot in it. This is a circle with a dot in it, and now we come to branches. Cool? Then, now we have to follow this other. Okay, we know it comes out, we know it branches again. What did I use for that? A squiggle. I was running out of ideas. Maybe use a black squiggle. Squiggle. Okay. This branch point is a squiggle. We've got a squiggle. One of those lineages goes all the way up to the top. All the way up to the top. Another one comes up to another branch point. I apologize that these are running into each other. We come to another branch point. And that final branch point, we're going to do a yellow square. Final branch point is represented by a yellow square. Come up and up like that. That's how this tree over here can be made into a rectangular tree, how those two correspond to each other. Okay, now discussing, coming back to how phylogenetic trees, we've talked about this with the squirrels, how they can be used to figure out when things happen. Looking at primates, humans, we do not have an opposable great toe. Our big toe is not opposable. We can't like grab things with it. My husband jokes that I can because I have crazy long toes. But technically, our great toe is not opposable. And this is a like really, really simple example of trying to figure out when did that happen. If we put that transition happening here, well, this ancestor has an opposable great toe, as indicated by this here. If the opposable toe was lost here, I'm not sure if that's the right spelling for that, was lost here, then we would have to have a transition here back to the opposable toe. And then this lineage leading to humans would retain the non-opposable toe. Makes more sense if that change happened out here between the lineage of this ancestor and humans. Just a demonstration of how to logically think through using a phylogenetic tree. But in order to do that, we have to have the tree. Okay. So, another concept when we're talking about phylogenetic trees, when we're talking about evolutionary relationships, and I hinted at this earlier, we've talked about homology or gene orthologs, so things that are related by common descent. Just because things are similar doesn't mean that they're related by common descent. We saw that with the shark and with the dolphin. If we look at these two species of birds, their most recent common ancestor does not have this elongated beak. So they didn't inherit that. They didn't get that by vertical transmission. They independently, two different lineages, adapted that long beak. And it's because both of these species of birds feed on these flowers that have these very long tubular flowers. So to get into the nectar, they need that long beak. So at some point in their evolutionary history, the evolutionary history leading to this species, there was a bird with a slightly longer beak. They were able to reproduce better. They had higher fitness. So they gave rise to birds with longer beaks, which gave rise to birds with longer beaks, which gave rise to birds with longer beaks. But each of these species, they don't share a most recent common ancestor. So this is what this is called, convergent evolution, where the environmental pressures cause two separate lineages to converge on the same solution. So they both face a similar quote-unquote problem, they converge on a similar or the same solution. That's convergent evolution. Another like wicked cool example of convergent evolution is the octopus eye and the mammal eye. Our common ancestor, which you have to go back pretty stinking far, didn't have an eye. Each of these lineages evolved eyes independently, convergent evolution. And if you look at the anatomy and the physiology of the eye, that becomes kind of obvious. We tend to think of our eyes as these really exceptional, like, masterpieces, which they are. But this is an example of macroevolution constraining microevolution when it comes to humans. So light is coming in from this side, this angle. This is light. And in the octopus eye, I wish I had gray. These structures right here are the photoreceptors. So light comes in, interacts with the photoreceptors, and then the neurons carry that information toward the brain. Makes sense, right? Our eyes, not so much. Here's light, right here, coming in. Okay, cool. Our photoreceptors, way back here. Do you see all this mess that light has to pass through to get to our photoreceptors? Light has to pass through all of these cells, all of these specialized neurons that are carrying information, because where it goes to the brain is from here, the front of the retina, like in front of the photoreceptors. Doesn't really make sense, right? Well, this is an example of convergent evolution. Our eyes are functionally quite similar, but microevolution had different stuff to work with to come to that convergent solution. So in the case of the mammal eye, macroevolution, the ancestor from which this eye evolved, didn't have the same physiological anatomical structure that the octopus had to work with. So microevolution was constrained by its history, by macroevolution. Okay? And so the octopus eye and the mammal eye converged on essentially the same solution. But it looks very different because of what evolution, the macroevolution, the history of the evolution, the background on which the eye evolved. This is just an image to show you that evolution is typically pretty slow, graded. You can graded evolution. These different sandpipers. So this one has a relatively short beak, a little bit longer beak, a little bit longer beak. Whoa, that sucker's long. So we see gradations of that, of the evolution, of that adaptation. Oh, there should be something else there. Tells me it's the end of the show. Thought there was another thing. Okay, so those are sort of the basic concepts that we're thinking about when we're thinking about evolutionary trees and looking at structures, using structures. We got to think about homology. We got to think about, is it, when we're looking at genes, is it orthologous or paralogous? Graded evolution. Are we talking about convergent evolution? All of these different things come together and are part of figuring out an evolutionary tree. All right, I will see you back here next time.